Hello, my name's Ian Stewart, and I'm really glad that you can join me for the second part of a four-part series on Introduction to Problems of Evolution. So in the first talk we looked at what evolution is. We also looked at various problems associated with the belief that material plus energy plus time could have resulted in a biological organism, the first biological organism just arriving, or being produced through non-intelligent process. Basically looked at three major categories of reasons for that. First is obtaining the necessary molecules, the protein and RNA molecules, and we looked at the difficulty with obtaining them. Secondly is obtaining the cellular structures. And thirdly was obtaining the code, which is the terminal argument. So the fact that a particular sequence represents a particular set of instructions, what is being transferred through there is a non-physical message. It's got nothing to do with material or energy. In fact, if it was the result of material and energy, the code would be break down into predictable patterns. Biological life, that first biological organism, would not have arisen without an intelligent agent. In the second talk, though, we're going to be looking at genetic variation and the role of mutation in relation to genetic variation. And we're going to just assume for now that first biological organism did actually just arise. We're going to, as it were, waive the problems which we noted in the first talk, and we're going to say, well, what if that first biological organism did just arrive? And it's perhaps worth just noting that a single-celled biological organism is not something which is incredibly simple. Guys who were around in times of Darwin and guys who followed him, guys like Huxley, guys like Haeckel, used to believe that biological life, single-cell life, was very simple, that it was some sort of undifferentiated globule. But if we look at a single-celled biological organism here, you can see that it has the same essential role for, say, DNA. The information storage retrieval system is the same. Messenger RNA, you've got the flagellum motor there, cell wall, capsule. The ribosomes are the same. It's the protein building factories. A lot of the proteins have the same functions. So it is extremely complex. It isn't just the transition from non-life to life is not sort of watery. It's quite fundamental, so that's worth noting. But if that first biological life, something like that, did just arise, I should actually perhaps just mention that some Darwinists may say, well, that's a modern single-celled organism. So in the past, single-celled organisms were much simpler, so perhaps it was easier to achieve than this single-celled biological organism here. Well, I think what I'd say about that is we basically know that isn't true. And the reason we know that isn't true is because the reason that we can identify that biological life was around, say, 3.5 billion years ago is because we observe stromatolites. Stromatolites are structures which we observe in rock 3.5 billion years ago, which is produced by the action of bacteria. Now we know those structures are produced by the action of bacteria because we also have stromatolites forming today produced by bacteria. So you can't say they're fundamentally different bacteria then were then than they are today, because if you look at the structures they're producing, if they're actually essentially identical, then it is difficult to argue that they were fundamentally different in the past. So that first biological organism, though, if it did arise, it would be a single haploid genome, one copy, and it would basically, if we think about it, how would we define whether that first biological organism was alive? And many people would say, well, that first biological organism would have been able to replicate. Now what that organism would be able to produce, what it can make, would basically be dedicated by its genes. It couldn't do anything if it didn't have the code. Now if there was no change to its genes, if there was no fundamental change in the genes of that biological organism, according to Darwinism, then after 3.5 billion years, biological life would basically look like this. It would all be identical to that first biological organism if there was no mutation, if there was no variation. Now the one thing which prevents that from happening, the thing which will be able to convert that single-celled organism to the human brain. Now the human brain has about 86 to 100 billion neurons and about 100 trillion synapses. Okay, extraordinarily complex. The one thing that will get you from that to that, okay, is actually mutation. We look at an example of mutation occurring. You can see that the C has become a T. We should note that when you get a third nucleotide, the bases are read in 3-letter codons. If you get the alteration to the third nucleotide base, it doesn't always affect the actual amino acid. So given the fact that mutations are highly significant in converting, it's going to be decisive in converting that first biological organism into, say, us, you would think to yourself that mutations must surely, if we discover them as the ultimate source of all genetic variation, which they are, they must surely display tremendous beneficial and innovative effects. The reality is, though, when we do look at the role of mutations, they are nearly all neutral or bad. I mean, the vast majority. In fact, Mohammed Noor of Duke's University actually compared a beneficial mutation to throwing a hammer at a car. What are the chances of beneficial mutation? Okay, it's not zero, it's not nothing, but it's extraordinarily unlikely, okay, because biological life is streamlined. So if you introduce randomness, then it's going to have negative effects. A lot of diseases are obviously associated with mutations. Now, Behe wrote The Edge of Evolution, which looked at the mutations, the role of mutations in more detail in relation to evolutionary theory, and he studied it in relation to malarial resistance. Okay, so malaria did experience a mutation where it shift in 2 amino acids, okay, which gave it resistance to the drug chloroquine. Now this is extraordinarily unlikely. It's actually incredible that it actually happened. Okay, now it happened because you basically have about 1 billion people a year get malaria, and in each infected person you have about 1 trillion infected cells. So because of the huge numbers involved, it was actually possible for this mutation to occur. Now he called that a triple C mutation, a CCC cluster. Okay. Now for that to occur in a much smaller human population, he calculated would not be plausible, even considering many times the duration of the Earth. In terms of the entire age of the Earth, it would not be plausible for that to occur in a human population. So if you consider trying to convert an ape into a human, 7 million year time frame, you know, it's extremely difficult. Okay. Now for a mutation of twice the complexity to occur in any biological organism, or just occur at all in any successive generations, including in the whole of biological life, would not be plausible even granted the entire duration of the Earth. All of biological life, it would not have something that is twice as complex as the mutation which gave rise to malaria's resistance to chloroquine. Now, if we do observe structures in biological life which is bursting with such examples, where it would require many times the code would have to be altered, you know, to much greater extents than just shifting 2 amino acids, then yeah, we can conclude that an intelligent agent was involved in that structural change. That is reasonable to suppose. He also looked at, say, E. coli, gone through, say, 30,000 generations, nearly 40,000 generations of E. coli, and we do observe natural selection, the struggle for life. What we're generally finding, though, is that the trend is downwards. Okay, it's like trench warfare. Okay, they start to make themselves more efficient in the struggle for life. They start throwing away chunks of their genetic patrimony because it conserves energy. It starts losing its ability to make certain RNA bases, for example. So, that being the case, it is interesting to note that the laboratory experiments in relation to the role of mutation in bacteria is actually consistent. You'll notice that when Darwinists are giving arguments trying to explain why evolution is true, what you'll generally find is they'll start referring to something that's gone wrong. Okay, so they'll look at the vestigial argument. Okay, so the grey whale used to have hind limbs, now it does not. It's lost something. What about the human appendix? It's lost function. Okay, there's a lot of or atavisms. Look about the snake. It used to have legs, but now it's lost those legs. Okay, a lot of the major arguments are loss of something, and that is consistent with observations about the role of mutation. Now we'll also look at Stephen Meyer's article on uh, um, a piece for a peer-reviewed journal, The Origin of Biological Information in Higher Taxonomic Categories, which is quite a famous article. And what he basically did is he did not produce new arguments, he just systematized arguments that had already been made and pieces of evidence which had already been published in peer-reviewed articles. He basically systematized them. And he showed that, say, there's a particular protein which is 400 amino acids in length, which Cambrian animals would require, and in order to build their stout body structures. And it was basically noted that to arrive at the correct sequences to code for this protein was not actually plausible, even granted the entire duration of the Earth. That if you are trying to obtain new genes from coding sections, if you get alterations in those coding regions, because the number of active nucleotide sequences which give rise to actual proteins is so rare, when you get an alteration of a coding region, then what you get is basically loss of function. If you get non-coding sections, it's akin to finding a very tiny needle in a very, very, very large haystack, trying to find, through altering nucleotide sequences, a active sequence which actually gives rise to a particular protein. And also just new body plans. If you've got to take say, to convert an ape into a human, it's not just a question of getting a new protein, which is going to be a challenge anyway, or converting an existing protein. You're going to need, new cell types. You're going to need many different proteins occurring at the same time. You're going to need not just new cell types, which you're going to have to build, but you're also going to need new systems of protein to service those cell types. You're going to need many new cell types together to get the new tissue. And that's not even considering new sort of body plans, adaptation of pelvis and so on. And there's also there's significant problems which were noted there. Lots of slander about the lead reviewers, but they couldn't argue with the actual arguments, because they were all peer-reviewed arguments. Now, in relation to new protein functions, I won't get into that because it's quite an involved subject, but basically, if you have one protein fulfilling a particular function, to convert that protein into, through sort of mutation, to convert a protein from one function to another, the number of alterations and sequences which have to occur is not plausible. That's another major problem with the theory of evolution. So in summary, we've basically looked at single-celled life, and we've noted that it isn't plausible for single-cell life just to produce multicellular life, at least on the basis of the evidence we have at the minute. Studies of E. coli, studies of malaria. It's never been observed that a population of bacteria could collaborate in such a way, or one ingesting another, to produce a multicellular organism. We've also looked at multicellular life, and we looked at the problems of obtaining the new genes and converting existing proteins to perform new functions. We just mentioned that briefly. Problems with obtaining new cell types, problems with obtaining new tissue. So we concluded that mutation is basically insufficient for converting a single biological organism, single-celled organism. If you did have that initially and you had lots of mutations, what you basically end up with is a worse version, most likely, of that original organism. You certainly would not end up with the human brain. So in conclusion, it's just worth reviewing quickly some of the things which a ape would be required to do. Say if you had Australopithecines and you wanted to convert them into humans, well, if you've got you would require a new skull, you require a new spine, expansion of brain, adaptation of pelvis, adaptation of knees, adaptation of feet, different jaws, alteration of muscle attachments, relocation of sinuses. So, I mean, there's a problem just obtaining one protein. The sort of structural changes which are required, it's not plausible. If you look at an ape there and a human being, I mean, what you generally find is not a lot of detail. Darwinists don't really tend to look into the detail, but is it possible for the nucleotide sequences of, say, an Australopithecine to convert into a human? It's generally just comparisons of nucleotide sequences rather than anything any more detailed. So A Pocket Guide to Problems of Evolution is written by me. You can download it on Kindle, which gives you more information on this. And that's basically the end of this second talk, so I look forward to seeing you again for the third and fourth talk. Okay, thank you.